continuation of the previous video that I posted, which was about planning an investigation question, where I gave you an acronym that was Mrs. CID. And I told you what the details of Mrs. CID, each letter in the acronym stands for. M for method, R for repeat twice, S for safety, C for control variables, I for independent variable, D for dependent variable. So if you want to check the details of how we use Mrs. CID, you can check my previous video. In this video, I'm going to explain how to use this acronym for an exact question that I'm going to solve in front of you and give you an idea about how we should plan, how should we write our investigation answer to get full 6 marks. Okay, so according to this acronym, and I also told you the order in which we write. So this acronym is to remember the components of your planning investigation question. And I gave you another order in which we write these acronyms. So let's start. So first of all, you have to read the question carefully and you have to identify what is the independent variable and what is the dependent variable. So like in this case, the question is, they have given water moves into and out of cells by osmosis. Plan an investigation to determine the effect of. Okay, now in every question, you will always get this part where they say the effect of. Okay, so whatever comes after effect of, that is your independent variable. So here what they have written is effect of the concentration of sodium chloride solution. That means this is your independent variable. Okay, and the dependent variable is what follows. The rate of osmosis in plant tissue. Here they have just mentioned plant tissue. So generally what we take in, we can take for osmosis experiments is we can take potato cylinders of equal sizes. So to cut potato cylinders of equal sizes, we use an equipment which is called as cork borer. So I'll show you this cork borer. How it looks like this. So this is a cork borer. It's like a hollow metal straw, and we insert this cork borer into the potato cylinder after it has been peeled, and then we make such small cylinders, small or large depending upon our requirement. So basically, why we use a cork borer in cut cylinders, potato cylinders of equal dimensions, and then we can use a ruler and knife to cut them into equal lengths. And then, so that is how we will make sure that the size of the potato tissue that we are using in this experiment is same. Because osmosis, which is movement of water, will also depend upon the surface area. So we have to make sure that the surface area is same at the beginning. Okay, so when you're writing your investigation question, the first heading that you will write is your independent variable. Independent variable, you just put the heading as independent variable and write what it is. Okay, so like I told you already, it is in this case the concentration of sodium chloride solution. Okay, sodium chloride solution is your independent variable. Okay, then dependent variable. So what is our dependent variable is we need to understand the rate of osmosis, right? But to understand the rate of osmosis, there can be 3 different ways how we can do it. We can either measure the initial length and the final length, or we can measure the initial mass and the final mass, and so we can know if there is a change in mass and or a change in length, then we will know where osmosis has taken place fastest. Okay, so for now, let's take dependent variable will be as the initial and final length of potato cylinders. Okay, so how will we measure this initial and final length of the potato cylinders? Is using a ruler. Okay, that we will mention in our method. Now, control variables. So we have to mention at least 3 control variables. That is always because IGCSE mark scheme will give you anywhere between 2 to 3 marks for writing control variables. So it's always better to write 3. So any factor that can affect the rate of osmosis in the cells has to be kept constant. We are changing the concentration of sodium chloride solution, but the volume of sodium chloride solution will be one of the factor that has to be controlled. Okay, so volume of sodium chloride solution. Then temperature is a factor that will affect the rate of osmosis. So we have to control the temperature using thermostatically controlled water bath. And one more thing, the initial length. Okay, the initial length of the potato cylinder should be same. That has to be controlled, as well as the the type of specimen that we have taken. Okay, so it's ideal if we take the same potato cylinder. Okay, so the type of plant tissue that we are taking. So in this case, the type of plant tissue that we are taking is the potato tubers. Okay, these are your control variables. Next thing we have to mention is a relevant safety precaution. Now, in this case, what we are going to use, is there any safety hazard that we are going to face? So, only one thing is that when you are cutting the cylinders of the desired length, you will be using knife, right? So, that could be a safety hazard. So, what care we have to take when we are handling knife is use a flat surface for cutting the potato cylinders. Okay, so this is a safety precaution, and that is why we use white tile for cutting the potato cylinders. Next, we have to write the method. Okay, now what the method would be, how we start is we will take 3 test tubes. Ideally, we should, like IGCSE will allow you to mention anywhere like a minimum of 2 is required. More than 2 is fine. Okay, so I generally suggest my children to write 3 different setups. So the first part of the method would be we take 3 test tubes, okay, and then we add equal volume of sodium chloride solution in each. Wherever possible, you can mention the numerical value. If instead of writing equal volume, or you want to write that in bracket, let's say 10 cubic centimeter. Okay, that we are writing in bracket. So we are giving an exact value. Add equal volume of NaCl solution to each, full stop. And then what we do is add 3 potato cylinders to each test tube, full stop. Immediately start the timer. And after that, how long do you think? What should be a reasonable time length for which we should keep the cylinders dipped so that a substantial amount of water loss or water gain will take place, and it will allow us to see a measurable difference in the length or mass. Okay, so you can mention anywhere like around 30 minutes should be a good time, or 20 minutes is also fine. Right, so immediately start the timer, keep the cylinders, keep the setups at 37 degrees Celsius using thermostatically controlled water bath. Wherever possible, we have to mention the apparatus or the equipment. So using thermostatically controlled water bath, keep the setup at 37 degrees Celsius using thermostatically controlled water bath for a total duration of 30 minutes. Okay, full stop. After 30 minutes, then what will you do? You have to after 30 minutes, remove the cylinders and measure the final length using a ruler. Okay, so this is your method. And after, so first initially we have measured the initial length, we have kept same, and then we measured the final length. Okay, so that is all that you need to give in your method. And after that, you have given a safety precaution. The final thing, let's see in Mrs. CID, what all have we covered? We covered the method, we covered safety precaution, control variables, independent and dependent. Now only repeat. So in repeat, what you need to write is, this is a standard line which you will always write and you will always get 1 mark on this. Okay, so repeat the entire investigation twice to identify the anomalous result. To identify the anomalous result. Okay, this is all that you need to write in your investigation answer. Now let's check with the mark scheme how many of our marking points are there matching with the mark scheme. Okay, so what did we write here? Independent variable, we wrote this, 2 different concentrations of sodium chloride solution. We have mentioned this. Then details of measuring dependent variable. So how will we measure the dependent variable? Cutting cylinders of potato tissue. Leave cylinders into sodium chloride for suitable length. This we have written. Measure the length before and after immersion. This we have written. Extra details of surface dry the tissue. This is required only when we are measuring the mass. Okay, so for now this is not required. Cutting cylinders of plant tissue using a cork borer. This I told you we have to mention this. Okay, and then they are accepting 2 control variables. So let's see which one did we mention. We mentioned the temperature, we mentioned immersion time, we mentioned the type of tissue, we mentioned the initial length and volume of solution. So we have mentioned 5 control variables, but according to this mark scheme, we can still get a maximum of 2 marks here. Okay, 2 or more additional repeats, we wrote this, and then we gave the safety precaution, which is cutting on a flat surface. Okay, so total how many marking points have we covered? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. 11 marking points you have covered if you follow the acronym Mrs. CID. So you will definitely get a 6 on 6 if you use this trick to write this planning question. Hope you understood how to answer the planning question. You still have any doubt in how to approach this plan the investigation question? Please write in the comment section. I will reply.